Okay. Good morning, good afternoon, and good evening, everyone, joining us from around the globe. Welcome to FABI International Seminar Series. I am Neriman Yilmaz, and I'm a postdoctoral fellow here at FABI. And today I will host a seminar for this session. And in the boardroom, I am accompanied by Professor Bernard Slipper and Dr. Marcus Wilken, who are also hosting the seminar together with me. As you know that we are already more than 100 people here, but it doesn't matter really who you are or where you are from. One thing we have in common is that since December 2019, COVID-19 affected all of us. Some of us either got COVID by ourselves, or we lost someone that we love. And during this time, many countries had their own struggles. One of the most recent and the reason why we are all here is the COVID-19 associated mucomycosis happening in India, and also which is publicly known as the black fungus infection. So once this problem occurred, people immediately started to ask the question like, what is mucomycosis? Why it is happening and why is it associated with COVID-19? Why it is happening in India? How can we pretend it? How can we diagnose it? And is there any way to treat it? And luckily today we have a world expert speaker with us, Professor Arunaloke Chakrabarti, who can answer all those questions with even extra information. Professor Chakrabarti is the head of Department of Medical Microbiology at Postgraduate Institute of Medical Education and Research, Chandigarh, India. He is also the head of National Mycology Reference Laboratory of India. He leads the Indian National Culture Collection of Pathogenic Fungi, and he is the president of International Society of Human and Animal Mycology and a coordinator of two Isham working groups. His major area of interest are in the field of fungal rhinosinusitis and mucormycosis, and also the allergic lung infections. Professor Chakrabarti, thank you so much for joining us today. We are very pleased to have you here to present to us, and we are looking forward to your presentation.
Thank you. Thank you very much, the FABI, as well as Dr. Nariman, for giving me this opportunity. And good morning, good afternoon, good evening. This is one of the opportunities which gave me the chance of meeting like person whom I have not seen for a long time, like Willan, Alessandro, all these people, they are at such a distance. Okay, the topic which have been given to me is very challenging one, because till today we are facing the same problem. The case numbers have come down, but what we are facing now is the cases with recurrence. Friends, let's start with an introduction in the topic of mucormycosis, and then I will go into what exactly happened in our country. We generally say mucorales are everywhere, whether you go to Antarctica or you go to having hot geothermal soil where you can see mucor. Out of these 55 genera, 261 species, 38 species are human pathogen, of which Rhizopus aereus is the commonest one. Generally, we see the species under Rhizopus, Rhizomucor, Lichthymia, Mucor. These are the common species causing human infection. But there are rare species, especially like Apophysomyces, Succiniana, even Lichthymia, even in case of sometimes we see Mucor irregularis. Interestingly, this rare species, though Apophysomyces has been isolated in Australia and many other countries, even in USA, but I would say that 50% of all Apophysomyces cases are from India. So there are rare species which are geographically restricted much commonly in Asia, especially in India. Let me give some introduction to this disease of mucormycosis. The mucormycosis is usually classified on the basis of the site of infection. And very interestingly, each site of infection has got particular risk factor. Why and what are the reasons, still not clear. But like rhinocerebral disease or rhino-orbitocerebral disease, where uncontrolled diabetes is the risk factor, Whereas in case of the pulmonary disease, it is the hematologic malignancy or transplant recipients. Gastrointestinal mucormycosis, which we see in case of premature babies, as well as patients with malnutrition. Cutaneous group are seen much commonly in case of the trauma patients or burn patient, and of course, disseminated variety in immunosuppressed patients. These are the different series which have been published across this globe. If you see, each of these clinical types have been given with a color. Like this blue color represents the rhino-orbitocerebral type. If you see in case of the European or in case of US, commonly we see especially the first three colors, blue, then red is of pulmonary, and the green is of cutaneous, almost equal in proportion. In some places, of course, pulmonary is more common. But if you see in India, India has very interestingly, we see more commonly the rhino-orbitocerebral type. Is it we are missing the pulmonary type? I'm not very sure, but there are increasing number of cases of pulmonary mucormycosis, but sheer number of this rhino-orbitocerebral type overshadows all other clinical types. So if you see in India, we see rhino-orbitocerebral type in around 65 to 70% cases, pulmonary 10 to 15%, cutaneous 10 to 15%, gastrointestinal 2 to 5%, and there is a very peculiar disease which we have found in India and to a certain extent in China that happens in case of the healthy young individual. Suddenly they suffer from this disease which we call isolated renal variety, that is around 3 to 5%, and disseminated variety is around 3 to 10%. Friends, in cutaneous variety, mainly it occurs after the roadside accident. But in India, what we have found that in case of the, especially in villages, where some quacks or those who are not having sufficient knowledge in medicine, even if they have tried with intramuscular injection, they're creating these necrotizing fasciitis cases. This is very disturbing because people suffer from this, even die from this infection. Nosocomial mucormycosis had been seen around 9% of the cases. But this particular disease, which I was trying to tell you, that renal mucormycosis in healthy host, patients suddenly develop fever, flank pain, hematuria or anuria, means there is blood in the urine, or even there is stoppage of the urine, and pathology shows infected kidney. Imaging shows this infarction, and you can see in histopathology like this presentation. And what we have seen is that over the last 2 decades at our center itself, we have seen more than 50 cases. And what is interesting is that because of the awareness, most of the cases are diagnosed ante-mortem. But if you can see, the mortality is around 50%, because when kidney gets involved, especially bilateral involvement, that's very difficult. The patient, if you cannot do the transplant, then the patient usually dies. Why we are concerned about mucormycosis? Generally, we say that this particular disease is fungal emergency. If you don't manage quickly, most of the cases, if it goes into angioinvasion, then there is problem. Second important thing is that if you have to diagnose, there is no biomarker test available. And in case of critically ill patient, you need to distinguish from aspergillosis, because the question comes, shall I prescribe voriconazole or amphotericin B? I would say that particularly this mucormycosis, not only in India, the incidence is increasing globally. But if you compare with any of the Western world, in India, number of cases, I'm saying all this before this COVID pandemic happened, even then there is around 70 times across the India, we have seen the cases of mucormycosis. You can see in our center how the case numbers had increased before this COVID. Not only in immunosuppressed, we have also seen ICU the mucomycosis. This study, which we have published about 2 years back, where it had shown that this is 11 ICU study, where we have shown that mucomycosis out of invasive mold infection was around 24%. And diabetes is the most common risk factor in this group. So friends, diabetes, when we try to look into underlying risk factor, again comparing to the different series published, in India, if you see here the diabetes shown by blue color, which shows very high in incidence. What is the reason? The main factor is that we love sweets. And we are really hesitant to do our blood sugar estimation. That's the reason when we have done this different series published, we found that considerable number of patients, they are ignorant about their underlying diabetes. You know, if I diagnose any patient with hypertension, he has to take the medicine throughout life, but they don't mind about it. But if you diagnose a case of diabetes, then there is problem because people are very scared that they cannot get the sweets. So what we did long time back, a study in consecutive diabetic patient, over 22,000, we found 35 cases. So you can see, imagine such high incidence. Out of 1,000 diabetic, it is 1.6 cases. And India and China are the two leaders in case of diabetes in the world. Now let's go into the COVID-associated mucormycosis. I'll start the story since last year because the way it had evolved, it would give you the clear picture how this COVID-associated mucormycosis had come to India. You know, last year at the beginning when we started having these COVID cases, we found there are occasional cases. But suddenly there was a series published from one city, from 3 eye centers, 18 cases that had really raised our antenna. And we started looking into what is the picture across the globe. There are two systematic analyses have been published. One from India, one other is a combined ISHAM and ECMM together had conducted this study. What we found is that India has the highest number of cases during this period. And if you see, uncontrolled diabetes and systemic steroid are the major predisposing factor, and rhinos orbital-cerebral mucormycosis was the most common presentation. And mortality between these two series, one series reported 31%, another is the 50%. This is just the systematic analysis of the cases which have been reported. At the same time, this was one of the cases which we have seen uncontrolled diabetic patient post-COVID, 55 years old, suddenly had cough and expectoration. And you can see when you did the CT, there was a big cavity which had been seen. And the sputum was positive for rhizopus microsporus. And this patient was diagnosed as probable mucormycosis, treated with liposomal amphotericin B, then posaconazole. And when we tried to review all the cases of COVID-associated pulmonary mucormycosis, we found there are 5 cases, and 2 of them have been reported post-mortem. And here, mostly diabetes, the ARDS or renal compromise are the major complicating factor. One of the 5 cases, both rhinoorbital and pulmonary mucormycosis were there. So it raised our antenna. We felt that we should do one study. So this is the study which we have published in Emerging Infectious Disease. This study we had conducted during September to December 2020, last year. 16 center study. And this 16 center study first showed that compared to 2019, there was a nearly 2.1-fold rise in number of mucormycosis. What we found very interestingly that it is the male who suffer more commonly and the older patient. Then we tried to look into what is the exact prevalence of COVID-associated mucormycosis. At that time, I'm saying all the story of the last year. Amongst all the COVID patients treated in those 16 centers, it was around 0.27%, quite high number. And those who are treated in ICU, it was 1.6%. Majority of the cases were nasoorbital mucormycosis, and you can see nearly 1/4 of this patient already brain involved. That means the patient reported quite late to the hospital with this mucormycosis. What we found is that diabetes was in 2/3 of the cases, but 1/3 of the cases, there was no diabetes, but the patient received steroid, and COVID was only underlying disease in those two. This particular data is very important, which is now helping us much more. What we found, majority of them diagnosed at least after 8 days after COVID diagnosis, and median period is 18 days. So you can see this waterfall figure. It shows that this is 18 days. That means these patients are already discharged from the COVID hospital. They have gone home. So here the question comes that if we have to diagnose early, we have to train them what are the early symptoms and signs. This was the very important finding which we found. Multivariate analysis found that hypoxia and inappropriate steroid was the very important risk factor. I would detail out this inappropriate steroid because that's very important. At least in these 60%, half of them, they were not having any hypoxia. But in spite of that, they have been given steroid. And other half, what happened is that instead of 6 milligram dexamethasone, which is being recommended by WHO for 10 to 15 days, they have been given very high dose and even for a more number of days. So following our study, this is another study from India came, that is January to March 2021. This was in 3 tertiary care centers. Their findings are exactly what we have found. It is they have got maximum cases which have got oxygen desaturation, and patients were in ICU, and we had our in our ICU 1.6%. Here it is 1.8%. Instead of 18 days, here after COVID diagnosis, 12 days they found. Orbital invasion is quite common, and you see instead of about 25%, here also 20% brain involved. Again, male were common sufferer, they were diabetic, and also steroid therapy was much commonly being used. Ferritin level had gone up in this group of patients. So this is very important factor. Friends, completely changed following that period. You can see what happened. This was the December period when we studied. And this is the period in May to June 2020. Just imagine how the case numbers have increased. This fungus has been said as a black fungus. People say it as mucormycosis, but you can see so many cases have been reported. So not only in our national media, throughout the world, people got concerned what is happening in India. This is a mucormycosis storm over Indian sky. So first time, one of the fungal disease had become notifiable in India. And this time, we have seen people in media started branding fungus with different colors. Black fungus, white fungus, yellow fungus. I don't know what would come next. But this created more confusion. You know, at least I would say mucor is not black. Its clinical presentation where ester is formed, which is called black, or which look like black. But mucormycosis, compared to this Demetrius group of fungus, is not black in color. But people started calling it as a black fungal disease. Now, this is in August 3, 2021 figure. Government of India report shows 47,508 cases. Even in this website itself, you can see it is mentioned, it is very likely that actual figures are considerably higher than this. That is true. lot of people, though it's a notifiable, were not notifying. Rhinocerebral is the commonest one. You see here the age group where the commonest or maximum cases is the 45 to 60 years. 85.5% had COVID-19 in this mucomycosis. Diabetes was in 2/3 of this. Inappropriate steroid was quite common. Then came this particular paper of 2,826 patients of mucomycosis. This is mainly from the Ophthalmologic Society, this paper had come. And if you see, look into this picture. This is in India. This is showing as per the density of the color of red. You can see where the COVID cases is very high. Now, maximum COVID cases are seen here. This is the Kerala area. And if you see, mucor cases were also seen in this same state or same province, also some other province like Gujarat. And what it had been found that, again, is the male commonly sufferer. Diabetes is the major issue. Steroid is 87%. Post-COVID, usually their cases, 44% are more than 14 days period. Orbital was the commonest thing involved, and they had to do orbital exentation, 34%. Mortality, at least in their study, had not shown very high. But when we did this systematic analysis again, which is again published just few days back in our Mycopathologia, this is our study where it had shown that mortality was much higher when the patient is ICU admitted or mechanical ventilator treated. Other than rhinocerebral, pulmonary and disseminated variety has more or higher mortality. And when we compare the mortality of India with global reports during this period of time, global mortality was much higher, probably due to the predominance of pulmonary and disseminated variety. Our cases were more of rhinocerebral mucomycosis. So friends, now come to the main question. Why such high number of COVID-associated mucomycosis cases happen in India? There are myriad hypotheses. Many people said that high mucoral spores around garbage, construction makeshift COVID-19 facilities, or said contamination of the oxygen because of the oxygen crisis. Industrial oxygen has been used. Respiratory equipment, some people said humidifier water is responsible. Some said reused face mask is responsible. Some said zinc supplements is responsible. Again, just few days back, but none of these theories is clearly proven. Just few days back, August 22, this paper had come, where we tried to look how the zinc plays role in mucor. 8 isolates we tried in vitro study with zinc supplementation. We found in 4, there is a time and concentration dependent increase in mean fungal biomass with zinc was observed. But when you looked into the patient, we found that mean serum zinc concentration was below the reference range in all the subjects and was not significantly different between cases and control. So our study did not support the speculation that zinc supplementation contributed in pathogenesis of mucormycosis. Friends, then we also studied the hospital environment. This is a study. This is the first time you are seeing very preliminary data. We have not published this paper. You can see in different areas where we have studied about the environmental condition. What we found is that in case of the oxygen cylinder, very few oxygen cylinder in the port, we could see mucor. Even in humidifier water, humidifier reservoir, nothing is there. But what we found very importantly is the air conditioned vents where mucorals were there. And if you see, even with the rooms with HEPA filter, in the air, mucor is there. But when it is not HEPA filtered, 50% of the room has got high count of mucorals. And outdoor, that is environment, where also mucus was quite high. And what we found this is not published data, unpublished, where we found Rhizopus aereus is the commonest one, which we also isolated from the patient. So we could not really prove, at least the environment is the major deciding factor in case of mucormycosis. But yes, in case of the ear, both in the hospital and in case of the environment, there is a problem. But at least it is not the this oxygen cylinder, unused or used mask, which are not so much responsible, at least we found. But what we have learned from the COVID-associated cases from the first COVID-19 wave, that means it was our study which have been done in last September to December, as well as another study which was published in January to March. Both these studies have shown unrecognized and uncontrolled diabetes is the major risk factor, and indiscriminate use of steroid at high dose is the major risk factor. Friends, this particular study, which had just again had come very recent, this particular study had been conducted in a tertiary care hospital in the city Mumbai. And if you can remember this particular figure, where we have shown this particular province had got very high incidence of mucormycosis. What they have done from March 2020 to May 2021, they have treated so many patients of COVID-associated COVID cases. And out of this, more than 1,000 in ICU and rest other in the ward. And in case of these patients ICU, 915 received steroids and 1,417 had diabetes, very high diabetes comorbidity. What they have done, they have followed a very strict steroid protocol in their hospital. You see, they have just given the steroid within the limit and within the period, which is maximum within 7 days. A nurse-driven strict glycemic control regime was also followed to maintain the blood sugar between 140 to 180 through the admission in ICU, and was achieved consistently in 82% patients. Most important factor, no case of mucormycosis was reported during the stay in the hospital or during immediate outpatient department follow-up. So friends, this particular study has indirectly proven that there is the major issue is the diabetes and mucormycosis, diabetes and indiscriminate use of steroid. You see, this is a center where maximum number of COVID-associated mucormycosis have been reported. But in their hospital, there is not a single case have been reported. So what is the narration? What happened during this second wave where we have seen such a deluge of cases of mucormycosis? Friends, I try to put 4 factors here. One factor is, of course, diabetes, which I have said. I will discuss about the steroid issue a little more. We are not very sure how much COVID is causing the problem. And 4th factor, of course, environment, because the spore count is very high. Now, what happened during this second wave? There were so many patients in the hospital that the doctors had to see the patient in the corridor, even on the pavement outside the hospital. And during this time, they could not take care of the glycemic control. They were all trying to save the patient by having proper oxygen. But then there is this oxygen crisis was there. Because of the oxygen crisis, what happened is that the doctors went on increasing the steroid dose with the hope that they can save the patient. So instead of 6 milligram dexa per day, in certain patients, even 30 milligram dexa or 40 milligram dexa had been given. 30 milligram dexa means 200 milligram prednisolone. Can you imagine if you are giving for a month of 200 milligram prednisolone, the blood sugar has gone through roof. I have not seen such high blood sugar like 700, 800 in some of the patients. So that was the major issue which is there. So friends, what I try to see is that look at this environmental factor. This is our earlier study which we have published. We had shown that the outdoor air sample and also in hospital, the spore counts or mucorales spore counts is very high in India. Also during this time, construction of new hospital zones, restructuring of the existing wards can potentially increase the load of mucorales in the environment. So occurrence of mucorales across vast geographic area and even in non-hospitalized individual suggests that multiple other factors may be responsible. Now look into the agent factor. This factor, I'm not very sure, but if you see, fungi may acquire virulence factor over time. We have seen this in microevolution in Cryptococcus that is causing hypervirulence. Dr. Wielander is here. He is expert in this area. He will vouch on this aspect. Even in case of the fungi, there is genus-specific difference had been seen in case of mucorales. Whether mucorales have acquired hypervirulence factor contributing in the current epidemic needs to be studied, which we are studying at present. Host factor-wise, diabetes, glucocorticoid, I have said, but COVID possibly also playing some role in this. Friends, now try to build up the story between this diabetes, steroid, and COVID. Look at this diabetes high prevalence in India. Also, it had shown two studies very clearly that COVID-19 virus damaged the pancreatic beta cell. And because of this damage, the insulin production goes down. That also helps in increasing the blood sugar level. Steroid, of course, increases blood sugar. We know very well. Steroid also causes impairment of the neutrophil migration, which is very important in case of defense against mucorales. Patients are in stress, and this stress causing the stress-related hormone like cortisol and adrenaline coming, which is also causing hyperglycemia. So diabetes itself, COVID-19, steroid, and stress develops a hyperglycemic condition. This hyperglycemic condition is an inflammatory state. Even antiviral immunity to COVID-19 also potentiate this inflammation. So what happens during this time? There is an alteration in iron metabolism. We have seen hyperferritinemic syndrome, and excess intracellular iron releases reactive oxygen, which damages the tissue. So free iron comes in the circulation, and free iron is good food for mucorales. Also, this COVID causing endothelitis. This has been proven in several of the autopsies. Compared to the influenza, COVID-19 causes widespread vascular endothelial injury. You know, our friend Ibrahim had shown from Stanford that these are the two receptors which are important. One receptor is our endothelial cells is having, that is GRP78, that is the glucose-regulated protein receptor, and mucorales have got homologue, that is POTH receptor. So if I explain with this cartoon, what is happening is that with high glucose, free iron, siderophore, this is causing endoplasmic reticulum stress, and GRP78 is expressed. And here, when mucorales is coming, mucorales get chance to cause the cellular endothelial cell damage. And because of endothelial cell damage, it can enter the blood vessel, cause thrombus, and which is leading to ischemia and hypoxia. At the same time, we are trying to look into whether COVID-19 is itself causing damage to the anti-mucorale immunity or not. This was a very early paper from Wuhan, China, when COVID-19 infection started, they had shown low lymphocyte, monocyte, basophil, T cell decrease. They also have shown regulatory T cell going down, severe patient with cytokine storm. But T cell, compared to T cell, it's more important is the innate immunity in case of mucorales. At the same time, what I have seen is that macrophage activity is increasing in some of the papers. It's not explaining whether COVID-19 really disturbs antimucorale immunity or not. But this particular paper had shown that there is, in case of this COVID-19, post-COVID, there is immune dysregulation. And this immune dysregulation possibly disturbing innate immunity, which we are presently studying now. So friends, I don't have answer to all possibly your questions, but one thing is very clear that environmental high spore count is important. In healthy host, it is not producing the disease because of the innate immunity. But when there is COVID, when there is high glucocorticoids, when there is diabetic patient, when there is endotheliitis, there is chance of getting this COVID-associated mucomycosis. So what are the unknown areas still? We need to study whether COVID-19 is disturbing the mucor-specific immunity. We are not very sure what is the complete reason of this high incidence. We have possibly looked into certain factors which we have not been studying. We don't know whether the Indian population has got higher genetic susceptibility and whether there is hypervirulent rhizopus aeregeas present or not, which we are also studying. At least we have found industrial oxygen humidifier masks are not so important, but we are still trying to find out if there is any other environmental source. Now coming shortly for diagnosis and management, in case of diagnosis, rhinocerebral mucormycosis is much easier to diagnose. You can collect the sample by nasal endoscope and you can easily find out by calcofluor stain, and the growth of mucorales are not very difficult. You can go and identify. Here, what I like to stress is that of late, we are finding around 5% of the cases, both mucorales and aspergillus are responsible. But this is not so easy in pulmonary mucormycosis. The question is how to differentiate from COVID-associated pulmonary aspergillosis. If you can collect the BL sample, transbronchial biopsy or CT-guided biopsy, that would be best for diagnosis. But people are scared till now in COVID patient to touch the respiratory tract for collection of the sample. Imaging is not helping because what we have found is that in case of the mucorales, as well as in mucormycosis, as well as in case of kappa or COVID-associated pulmonary aspergillosis, there is this thick wall cavities are being present. But at least if there is repeated negative galactomannan, beta-D-glucan, or PCR against aspergillus, this suggests COVID-associated pulmonary mucormycosis. 2 cases only have been reported in India about gastrointestinal invasive mucormycosis in post-COVID patients. Both these cases are accidentally diagnosed during surgery. Otherwise, it was really difficult to diagnose these cases. This gastrointestinal mucormycosis in India is not very uncommon. We have reviewed the cases of gastrointestinal mucormycosis before this COVID era. We found that in case of the neonates, especially, this triad of shock, metabolic acidosis, and abdominal distension, whereas in case of the older children, it is exposure to the broad spectrum antibiotics, formula and spoon feeding. These are the more risk factors which have been there. So we are training people how to suspect COVID-associated mucormycosis. As I have told you that these cases are happening just after COVID diagnosis around 18 days. That means by this time, the patients are already at home. So how they can suspect they can have mucormycosis? We are training them by this process. If you have got nasal blockade, if you have got nasal congestion, nasal discharge, local pain, facial pain, numbness, swelling, think about it. Very interestingly, during this pandemic, we have found quite a number of cases maxillary teeth got involved. There is loosening of the maxillary teeth which have been found. But in case of the pulmonary mucormycosis, very difficult how they can suspect it. During this time when this mucormycosis is coming up, we have this serious crisis happen about amphotericin B. It was almost not available in most of the centers. Now, if you look into this antimucorales drug, we know that liposomal amphotericin B is the drug of choice or primary therapy. It overcomes the challenge of toxicity and penetration, but problem was it was not available. We look back to amphotericin B deoxycholate, but we need to know how we can use it because of the infusion-related toxicity as well as nephrotoxicity. Isavuconazole, that's a new drug which is already marketed in India, but within no time it is completely vanished from the market. We were not getting isavuconazole. Posaconazole is the only drug available at that time. So we look back to our different data, we found that itraconazole is one of the drugs which was never been recommended for mucorales, but our in vitro data showed that the strains were susceptible to itraconazole. So a lot of discussion happened, and then after discussing even with the MSG group and also European friends, ultimately we have recommended this process. We said that, yes, we need to do diabetes control, we need to reduce the steroid, we have to stop immunomodulators, and we need to do surgery as much as possible to remove the necrotic material. And regarding medical therapy, what we have to do is that, yes, liposomal amphotericin B is the first drug of choice. If it is not available, then we have recommended amphotericin B deoxycholate. While giving amphotericin B deoxycholate, we said that give slow infusion, we need pre-medication. We have said that sometimes before and after, if we can give a normal saline wash, that is also very helpful. When amphotericin B is also not available, we have recommended either isavuconazole or posaconazole injection. When none of this available, we have recommended even itraconazole. And of course, rest others are the same, which is there in case of the global guideline in mucormycosis. That means if there is stable disease, then isavuconazole and posaconazole. If there is progressive disease, increase the dose of amphotericin B or consider the adding of azole. And toxicity, if there is, then from choline shift to azole. This is in case of our ophthalmologic friend, they have published and they have highlighted this aspect. Their question is, can I save the eye? As nearly 85% of the COVID-associated mucormycosis patients have got rhinoorbital type and 72% had orbitis involved. What they have started doing is that in the medial orbital area, they try to do the resection if there is limited orbital involvement and preserve vision. Then they have given retrobulbar amphotericin B injection or irrigation. Initial encouraging result, but this approach is difficult decision, require multidisciplinary expertise and close monitoring. Orbital exenteration is wanted if there is extensive initial orbital involvement or worsening of the disease with attempted conservative management. Friends, during this time, we have got a lot of misinformation and misleading facts which we have seen. First, already I mentioned, people say mucorales is the black fungi. Next important thing is that a lot of questions we have to face. We say this disease is not contiguous. It doesn't spread from one person to another. But this education and teaching was very important. Next important, it was not spread through the oxygen or humidifier, water or mask. We also said no antifungal prophylaxis because the incidence amongst this COVID patient is not more than 10%. A lot of people, when not getting drugs, they have been treated with voriconazole, fluconazole. Vori, flu, echinocandin, 5-fluocytosine are not affecting against mucorales. We discourage combination of antifungal therapy because of the cost, and also there is not evidence supporting combination. Friends, because of the black fungus campaign, what happened? Onions having this black color, people were scared of taking onions. It may have mucor. Even in your refrigerator, if there is something like this black color, people were scared about it. So we have to train what is mucor. That media campaign of black fungus was a real problem. So at the last, how to prevent CAM? First thing is that a poorly controlled diabetes is the major issue. Good glycemic control is important. Systemic steroids should only be used in patients with hypoxemia. Blood sugar should be monitored. Oral steroids are contraindicated in patients with normal oxygen saturation on room air. Even then, people have given steroid in those cases. We said that as these mucorales or mucomycosis occurs after 18 days, when they are staying in the, they have gone back to their home. People generally wear masks when they are going out. They don't wear masks while at home. We suggested, especially those who are diabetic, please wear mask at least for a month. That was our suggestion, and we campaign amongst the doctors to suggest to their patient that at least use the mask. That is very important. And during discharge of the patient, advise about the early symptoms. So friends, summarizing what I tried to tell you is that COVID-associated mucormycosis should be suspected in diabetic patients. Steroids should not be given indiscriminately. Good glycemic control is required. Suspect the disease early. Older age, diabetic ICU admission, glucocorticoid therapy need to be cautious. Other than rhinocerebral, diagnosis in other sites is challenging, so we need to improve the molecular diagnosis. Early diagnosis, prompt therapy are essential to minimize mortality. But here the problem of the cost of the antifungal is so much. Earlier study had shown 25% of our patients, we could not complete therapy because of the cost of antifungal. At the end, I'd like to invite you for this Congress, hopefully and surely we'll hold it in September 2022. Please come to India. We'll be by that time surely get out of COVID. Thank you very much for your kind attention.
Thank you so much, Professor, for your very informative. Some parts are depressive, I must say, but some parts I see the hope there. But definitely quite informative presentation.